What are living stock collections? And why do we need so many different kinds? There are many types of biological collections that are used for teaching and research. Perhaps you've visited a natural history museum and saw the dinosaur fossils, or insects, or vertebrate specimens. This video will focus on collections of living organisms. Your family may have spent time enjoying an arboretum or a botanical garden, or maybe a zoo or an aquarium. These are just a few examples of the many types of collections of living organisms, which we scientists call living stock collections. These can be divided into three general categories based on how many different species they have and whether they're genetically modified or not. Biodiversity collections contain hundreds or even thousands of different species across a broad range of species. They are not genetically modified. Examples of biodiversity collections include many that are very familiar, including zoos, aquariums, botanical gardens, and arboretums. This also includes collections your family probably hasn't visited. Things like seed banks that preserve seeds of flowers, fruits, and vegetables, and their wild relatives. Scientists use the wild relatives of crops to breed better crops, such as increasing drought or pest resistance. There are also collections of living microbes, or single-celled organisms, such as these hundreds of different species of microalgae at the University of Texas at Austin. These have to be maintained as living cultures in flasks because they won't survive freezing. Each strain has to be moved to fresh media in a clean flask every 2 weeks. That's a lot of work. These algae have been used to discover new ways of making biodiesel using renewable algae oil instead of petroleum. Other single-celled organisms can be preserved in the frozen state, such as bacteria, yeasts, and molds. This specimen from the Phaff Yeast Culture Collection here at UC Davis has been used to study red pigments, yeast oil, biofuels, and for discovery of new yeast species. The second category is germplasm collections. Collections in this category contain a narrower range of species. They contain wild-type organisms, plus plants and animals that are bred using traditional breeding practices. For example, crop germplasm collections such as this vineyard preserve hundreds of different varieties of wild and domesticated grapes. This category also includes primate centers, which are used for many research areas, such as to develop better diagnosis and treatment of human diseases. Collections of bacteria, fungi, and viruses that infect plants are used to understand and protect against plant pathogens that can threaten our food supply. Collections of E. coli and other foodborne pathogens, such as this facility at Penn State, have been used to fight food poisoning outbreaks you may have heard about in the news, such as bacteria that contaminated spinach or hamburgers or green onions. If you like beer, bread, wine, or cheese, you'll like this category of collections. There are companies that sell starter cultures of bacteria, yeast, and molds to bakeries, breweries, and wineries and cheese factories. Some organisms are used for very specific research. This salamander, called axolotl, can regrow a leg, so it is used for studies of limb regeneration. A collection at the University of Kentucky maintains 9 different varieties of this salamander. The most specialized category is called genetic stock centers. These contain a large number of specimens that were all made in the lab by mutating one or a few parent organisms. Hundreds or even thousands of research labs around the world contribute strains and information to a central repository. The researchers work together, such as to sequence the genome and identify the function of each gene, and develop tools to edit the genome of that organism. These include collections of bacteria, fungi, plants, invertebrates, or vertebrates that are called model organisms. Why do we need so many different genetic stock centers? Because each organism has a special property that makes it useful for different kinds of discoveries. For example, the bread mold Neurospora crassa has been used to study classical genetics because the 8 spores produced during meiosis are neatly packaged in the order they're produced. In the 1940s, this species was used by Beadle and Tatum to discover that one gene produces one enzyme, and they received the Nobel Prize for that discovery. Here at UC Davis, we have a facility called the Mutant Mouse Research Resource Center, which maintains over 50,000 lines of lab mouse used for many different areas of research, especially human health. For example, there are mouse lines that mimic human diseases, such as diabetes, Alzheimer's, and even COVID-19. This little plant, called Arabidopsis, is used in many areas of biology, including plant genetics, because it grows quickly and produces hundreds of seeds. A collection at The Ohio State University distributes 500,000 seeds per year to thousands of research labs. Because the bacteria E. coli is small, grows super fast, reproducing every 20 to 30 minutes, it has been used for decades for important biology research, leading to over a dozen Nobel Prizes. The E. coli Genetic Stock Center at Yale University preserves and distributes thousands of E. coli lab strains. Our lives are impacted in so many ways by living stock collections, understanding the world around us, diagnosing and fighting diseases, developing renewable fuels, growing and protecting our food supply. The scientists that build and maintain these collections deserve our gratitude.